biology. A lot of people think it's processing big genomic data sets, sort of bioinformatics. And that's an important part of systems biology, but the cells in our bodies and our cell-cell communication, they integrate information across multiple scales. So there's the genome, RNA, and then proteins, protein activity states, metabolites. And now we can make high-throughput measurements on all of these scales of information flow in a cell. So what we try to do in systems biology is say, how does the cell integrate information across these scales? And in particular, we focus on protein activity states and the effects of those. So for example, a cell gets a growth factor that may induce it to shed other growth factors, activate proteases, make it move, make it live, make it die. So we try to integrate across all of these flows of information using many different kinds of mathematics to understand and predict cell behavior, particularly as it relates to therapeutics. So why do we need tissue engineering and systems biology together? If we have these fabulous 3D models of liver and other organs, why do we need systems biology? Well, it turns out that, as we know, people are different. And a huge topic in therapeutics now is what people refer to as stratified medicine or personalized medicine. We want to relate the kind of information you could measure in patients, things like their genomic data, but also their cytokine levels, the molecule cells use to talk to each other. We want to relate the kind of measurements you could make in a patient to the kinds of measurements we could make in our in vitro model. So that in vitro, in vivo correlation, it's more than about did the cells metabolize the drug. It's actually about understanding how the drug works. So we view the marriage of tissue engineering plus systems biology as an incredibly powerful marriage for the future of the whole drug development process, everywhere along the pipeline. Efficacy all the way out to safety and even up to clinical trials comes together in the marriage of these two things. And in our research programs at MIT that I participate in and direct. So a particularly fascinating application of the marriage of systems biology and tissue engineering is in breast cancer, and particularly the aggressive kinds of breast cancer that metastasize and kill patients. And I speak from experience here because I myself had triple-negative breast cancer and went through the very aggressive chemotherapy and radiation and surgery that patients like that get. And I came to appreciate how hard it is to develop new cancer drugs to treat patients. So a patient gets the chemotherapy to kill the tumor cells that have left the tumor and disseminated throughout the body. We have a very poor understanding of how the chemotherapy is acting in the liver, in the bone marrow, in the lungs. And in fact, for triple negative, which I had, it tends to go to the liver and the brain and the lungs, and we don't know why chemotherapy agents fail in a very significant percentage of these patients. Now, I'm one of the lucky ones, both because I survived triple negative breast cancer with a low now risk of recurrence, but also because in the Department of Biological Engineering at MIT, there's a tremendous group of faculty members already working on the systems biology of chemoresistance in many kinds of breast cancer and in triple negative. So we got together and we put together a program to build models in our liver of micrometastasis of triple negative. And we work with colleagues from the University of Pittsburgh, and we work under the aegis of the NIH Microphysiological Systems Program. And what we're building is, why does a tumor cell get quiescent in the liver? So a beautiful thing about our reactor system is that it's got the 3D structure, it's many of the features of liver tissue, and you can put cancer cells there and get them to quiet down, just as one of these silent metastases might do in a patient. So now we have a model that we can say, you give the patient chemotherapy, what is it that lets that cancer cell die or proliferate? And maybe what is it that might stimulate cells that are sitting there silently grow? So the so-called escape from dormancy may be due to an infection, it may be due to some systemic disease. Now we have a model to start really studying this that's a human model of liver with the human micrometastasis, and bringing in the folks who know how to mathematically look at the signaling between the cells all comes together to help us bring powerful new tools to this very unmet need in treating patients for whom there's no targeted therapies. we've had some early publications showing the feasibility of the model. We're very excited that we have an abstract accepted showing that showing what we think is the first evidence that you can quiesce these very aggressive tumor cell lines in vitro in the context of a 3D liver. So that's a huge advance, because these cells typically, when you put them in culture, continue to grow, and if they're in a tissue system and they're quiescent, now we can start to bring them in and out of that and really have a model. So that work is just now at the point of publication. It's been won an award at a conference and coming to publication, and we hope to have enormous amounts coming out on that front, as the team is very, very productive between Pittsburgh, MIT, and Draper to pull all the things from the devices to the biomaterials to the cell biology and systems biology to make this work.